Welcome to today's Scientist to Students presentation, Great Lakes Research and Sharing, hosted by the University of Wisconsin Sea Grant. Today's scientist is Dr. John Berges from the University of Wisconsin-Milwaukee Department of Biological Sciences and School of Freshwater Sciences. Today's teacher is Mark O'Neill from the Bayfield School District, Bayfield High School. He teaches biology and aquaponics. And I'm going to spotlight you too, and that means that your video of you should stay up when you're talking, so that people can see you depending on where you normally would have appeared in your in the preview thing.
Okay.
And that way, if students do turn on their video, we're not able to capture them, hopefully. So should be good.
Okay. Yes, we have in my teaching, we have a running discussion about students turning on their videos. So I think we have a couple students here already. And
yeah, if they're here and they want to turn on their audio and introduce themselves, that would be great. So I'll remove the spotlight just for a minute here.
I guess I'll start.
Thank you.
My name is Gavin Marks. I'm an 11th grader at Bayfield High School, and I like guitar. That's a simple hobby.
Ooh, cool.
Alexia, are you willing to go next?
Yeah. I'm Alexia. I'm a senior at Bayfield High School.
Thank you. And how about you, Andrea?
My name is Andrea, and I also go in a senior at Bayfield High School.
Thank you for joining us. We're still waiting, obviously, for Mr. O'Neill and the rest of your students, and so we'll go ahead and give them a few more minutes to join us, and then we'll kind of do an official introduction. It's nice to know a little bit about who's with us, though, so thank you for sharing.
You're welcome.
Hello, Mr. O'Neill.
Hello. Trying to make sure we're set up here.
Mom, you want me to get the light?
Yes. Are we still missing?
No, they look gone.
Oh, is she? Okay.
So we have 3 of your students online with us, and they have graciously introduced themselves while we were waiting for you to come on, and appreciate that.
Good. Which students do you have? I can't see the whole list.
I have Gavin and Andrea and Alexia.
Oh, good.
Mm-hmm.
Oh, and if you could give me permission to
Yep, you're
record.
coming on.
Oh, look at that.
We've got it recording right now, so we should be all set.
But I need to record it so I can show it in my class Thursday, or let the students.
Yeah, we'll send you the file, because we tested it out, and even if you hit record, it comes into our Zoom because we're the host.
Oh.
So we'll send you the file. I can post it without the closed captioning for you.
Oh, perfect. That was the delay, wasn't it? That was the problem.
Mm-hmm.
Okay, well, I have 5 students in the room here.
All right.
So that's
Are
the other half of the class, and we're ready to go.
Okay, so I will go ahead and introduce Dr. John Berges from the Freshwater School of Sciences at UW-Milwaukee. And I am just going to say very briefly, thank you on behalf of Wisconsin Sea Grant for joining us today for the Scientist to Student presentation series. And we do want this to be kind of interactive and questions. So if you do have questions along the way, please go ahead and use the chat if you're able, or just unmute yourself and ask the question, whichever you prefer. And with that, I'll turn it over to Dr. Berges.
And actually with that, I think I'm going to have my students join so they can interact easier. So if you guys want to actually follow the link and join, but mute yourself while he's speaking, otherwise it's going to end up crazy. Sorry to interrupt that.
That's fine.
Yeah, I can't right now.
And I'm just going to turn off my video to make sure Dr. Berges has as much bandwidth as possible, but we should be good to go. So go ahead.
Okay, terrific. Well, excellent, folks. Thank you so much for the invitation. I'm going to talk a little bit about some of the work I do. And by way of introduction, I know I offered you guys the option of a couple different things I could talk about, and the one you chose was Life and Death in the Lake. That's the title I picked. And what I'm going to talk about in terms of life and death is a group of organisms I work with, the phytoplankton. So here's one pictured under the microscope in kind of a UV fluorescent picture. And these are the primary producers in the lake, the microscopic algae. And one group in particular I'm going to talk about are called the diatoms. I'll get to them in a minute. But to start with, by way of introduction, I'm a professor in both the biological sciences and the School of Freshwater Sciences at University of Wisconsin in Milwaukee. And professors, well, we are scientists, that's what we do, but we wear a couple different hats. And so one of the things I do in my daily life is I do research. That's about half my job. And so in my research, I'm just going to get control here. In my research, I work with a whole bunch of other scientists around the world. And here are some of my buddies. So this is a project I worked on almost a couple decades ago. And you can see we come from all walks of life. We've got people representing at least 6 countries here, from Australia to Britain to Spain. And we get together, we work on research projects, we have a lot of fun. So this job is absolutely great. Get to travel the world. I've currently got projects in Australia, another project in the United Kingdom. And so this is one heck of a fun job. And we ask questions about the way the world works. So what could be better than that? But I get to interact with my colleagues. We get to ask really interesting questions. Another thing I do as a scientist, as a professor, is I get to work with students. And my students are a diverse group as well. Pardon me. And so what you need to know about that is in the lab, who does the work? Well, I'm kind of a manager, and my students, they're the ones doing the heavy lifting. So I have graduate students, students working on research projects, doing their PhDs, doing their masters, but I have undergraduates in the lab doing their first degrees in a variety of subjects, including biology, and I get them in the lab too. So I'm an equal opportunity employer. I pull them into the lab, and students are critical. Because, hey, I'm getting older here. I'm running out of ideas, and ideas are what science runs on. So I am mining this group of enthusiastic young people in the lab because I need their ideas. So they come into the lab, we get the enthusiasm going, we get those ideas, and then I help them develop their skills, and with me, with each other, explore those ideas. And that is one of the most exciting things I do. So half of my work is research, half of my work is teaching, although a lot of that is teaching about research. And then the other half, yeah, 3 halves, I know, it's a busy job. The other half is doing all the sorts of administrative things, getting money to run my lab, doing all the paperwork associated with reporting on the research, the papers and the publications, the health and safety. I'm a bit like a small businessman in that my lab, I've set it up. The university gives me the space, it pays for the electricity, it pays for the water, keeps the heat on. But then if I want to do anything, I got to buy the equipment, I've got to pay the salaries of the people working there. And that means I need to run my lab, I need to run my small business. And that's kind of exciting. That's an interesting aspect of it too. So that's a bit about being a professor. So the organisms I want to tell you a bit about today, they're phytoplankton. They're primary producers in all of the Great Lakes, Lake Superior and Lake Michigan. And these guys I've pictured here are called diatoms. They're single-celled. Some of them are just big cells on their own. Others join up, and they're what we call colonial organisms. But they're major members of the phytoplankton in both fresh waters and in marine waters. And if you looked in the oceans, you'd see very similar pictures of these diatoms to what you do in the freshwater lakes as well. And the critical thing, and one of the most interesting things about them, is they make cell walls, they make their structure out of silica. Silica, as they use it, is more or less glass. And so if you ever in microbiology seen a petri dish, those little glass dishes in which we grow bacteria, well, that's pretty much the way they structure their cell walls. You've got little petri dish plates that fit together, and that's how they make their cell walls. So an organism making its cell wall out of glass, making a little glass house for itself. And they're pretty cool. So we know a lot about how these organisms live and grow in the lake, but one of the things that has interested me most, and for the longest time, is we know how they grow, but what happens to them after they're finished growing? Or in other words, when we look at an organism like this, what happens when it dies? What causes it to die? And so I got a question for you guys. You guys probably have been on this planet long enough and have a sense of living things. So tell me, what's going to cause a living thing to die? Suggestions? So no, I'm not going to call on anybody because I can't necessarily see you, but can you give me some ideas about what causes living things to die?
Predators.
Predators. Okay, so in other words, something can eat you, and that's going to finish you off, right? Good, cool. Other suggestions?
Leora in the chat says it's old.
Old? Okay, so we get old and die, huh? We age. Could a single cell age? That's a great question. I love that question. We're going to come back to that one too. Other thoughts?
Michael's typing. Good.
What do we have there? I'm sorry, I can't see the chat here.
Yeah, and I can see the chat, but I'm not sure where he may have typed it because it's not in our chat. So maybe he's tied into Mr. O'Neill's chat and not ours. So Mr. O'Neill may need to relay that to us.
Yeah, he said that the cell was unable to repair itself any longer.
Okay, so some sort of so the cell's got to grow and repair itself, and if it stops being able to repair itself, then it's going to die. Cool. We can think about some sort of deficiency, something it's lacking, and it's not going to be able to keep going. I like it. Other thoughts? What happens to things when you toss them in the lake? Sometimes they float, but what else could happen to them?
Think.
Yeah, so they could sink. And what happens if these cells sink to the bottom?
Is there a chance these things can drown?
Well, most of them are pretty well adapted to living in water, so they're probably not going to drown. But what could happen if you're on the bottom of a lake for a long time? Any sense of what happens? Other stuff falls on top of you.
Yes, and Mark has indicated less light.
Yeah, it's going to be dark down there. You're probably not going to be able to grow. You could run out of light. That's not going to be good. But if you think in geological terms, we make a lot of rock by sedimentation. And so we build up layers at the bottom of oceans and lakes. You could just go to the bottom and become part of the sediment. And you're not coming back from that. You become part of the sediment, part of the geological record. Oh, you're pretty much finished there. What else could happen? And I'm thinking particularly these days, there's something on our mind when we think about people dying. Mr. O'Neill's wearing a mask. Why is he doing that?
We heard diseases and viruses.
Perfect. So what about diseases? Okay, now in short, you guys are great. We've come up with most of the interesting ideas I was going to present. So let me go with that. I'm going to formalize that, and we can talk through some of these. But that's great. So when I think about death in phytoplankton or in diatoms, as a scientist, I'm thinking about these sorts of things. So they're going to sink. And if you sink, you get buried, you're done. I can think about a situation where you get eaten. Somebody eats you, well, that's pretty much an exit. I can think about getting diseases. I can think about cells maybe getting old and aging. I'm going to come back to that because that's a trickier one maybe. And then I can think of a situation where you've run out of something, and this idea of not being able to repair yourself, I think that could feed into, but this idea that you become deficient, that could lead to death too. And so what I want to do is explore these ideas. So let's first think about our food web, getting eaten. So here I have phytoplankton, including our diatoms here. They're eaten. And the things that principally eat the phytoplankton are called zooplankton. So phyto means plant. Zoo means animal. So plant-like things get eaten by animal-like things. Plankton, literally from the Greek, means wandering organisms. These guys are in the upper water. They get pushed around by the currents, so we think of them as wandering. And then the zooplankton get eaten by fish. That's a really simplistic food web here. So we think about this food web. Now, before we go any further, I just want to give you a sense of what's going on here in terms of sizes, because we think in the terrestrial environment, we're terrestrial organisms. Things are big in the terrestrial environment. They're actually quite small in the lake. But how small? I want to put this in perspective here, and here's how I'm going to do it. Anybody know? This has been in the news recently. What's this?
Got stuck in some canal.
Ah, yeah. So this vessel is called the Ever Given. It's one of the biggest freighters we've got, and it got stuck recently in the Suez Canal. You might remember that. It's huge. It's 400 meters. Anybody know the deep point in Lake Superior? Anybody tell me?
We've got a 1,313 feet, somebody said.
If you divide that roughly by 3, can you do that in meters?
450.
It's just a bit less than that, because we're talking meters instead of feet, but I'll tell you for free, you could take this vessel, and if you tried to sink it on its end, it would just barely go under the water in Lake Superior. Find the deepest point in Lake Superior, you can't sink this thing more than maybe your height below the water level. Think about that.
Okay, that's incredible.
Stunningly incredible. In fact, this is 90 meters wide. The average depth of Lake Superior, is just about 90 meters. So, wow, this thing is as wide as Lake Superior is deep on average. So that's shockingly big. No wonder this thing gets stuck in places. So that's 400 meters. So this is me. Now, I am actually not 2 meters tall. I'm actually only about 1.8 meters tall, but let's do a little rounding here because it makes the math easier. That would mean that this vessel is 200 times bigger than me. Okay, so it's 200 times me. Now, here's a zooplankton, and that is typically things that eats our little diatoms. That's 2 millimeters, which means from 2 meters to 2 millimeters long. That's 1,000 times. So this thing is actually 5 times smaller proportionately than I am compared to this freighter. Does that put it in perspective? I mean, we are talking super, super tiny stuff here. And then our diatom is typically, here's a pretty one. Our diatom is typically about 13 times smaller than our zooplankton. And this is a significant rule in aquatic systems. The things you eat are often about 10 times smaller than you. Now, my grandmother used to have this little saying. I don't know where she got it from, but she's from an old German family, and her expression was, never eat anything bigger than your head. I don't know what that means. She used to say it all the time, but if she were an aquatic organism, that would be pretty much right, okay? Aquatic organisms tend to eat things that are about 10 times smaller than them. So here it is illustrated. So look at that. Tiny, tiny organisms. So that's our size scale. And first and foremost, let's look at that. So we have these diatoms, they're getting eaten by zooplankton. What can they do to protect themselves? Well, the diatoms in particular, I mentioned they have these silica cell walls. And if you look at them under the right light, hey, look, you can kind of see the glassiness. Now, what do you think it's like to eat something with a glass covering? A lot of people have hypothesized the reason they do this is because it's protective. And so the question comes up, how much protection against this sort of grazer is it to have a glass cell wall? And we've debated this back and forth all over the place. How are you going to crunch into a glass covered thing? How are your mouth parts on this zooplankton going to do with it? And we discovered something interesting not so long ago. So here are the mouth parts, and they look pretty vicious, don't they? But we can do something called X-ray microanalysis. We can analyze these mouth parts and see what they're made of. And not only do these guys have protein in them, not only do they have some carbohydrate in them, but they also, here, this little colored analysis here shows that the very tips of these teeth, these zooplankton mouth parts, are hardened with silica. So the diatoms make their shells out of silica to protect themselves. The zooplankton harden their teeth with silica too. And is that effective? Well, I'm going to show you after they've grazed for a while, we can look at the diatoms. Pretty shattered, huh? So the diatoms may try and protect themselves with silica shells, but the zooplankton have tricks up their sleeve too. So it's a little bit of a battlefield out there, huh? Okay. How about getting diseases? Well, it turns out right now, we're worried about this little beast, COVID-19, and we've all heard of it. Let me introduce you to this beast, CPV-BQ1. That is a diatom virus. It infects diatoms. And how does it work? Well, all viruses fundamentally work like this. Here we have a healthy cross-section of a diatom cell. The virus attacks, it gets inside the cell, and what it basically does, kind of like a zombie here, it takes over the cell. And before, the cell had its own DNA instruction set, and the virus says, forget that. Whatever you were doing before, forget that. I'm taking over here, and what you will do now is nothing you were doing before. Now you are going to make virus. You're going to make more of me, the infecting virus. Here we have the cell filling up with viruses. And incidentally, when you're attacked by COVID-19, your cells start producing virus. And you don't feel very good when they start doing that, which is part of the illness. They fill up with the virus, and they fill up with the virus to the point where the whole cell eventually bursts. And that's end of story for this cell. Now I want to point out the size here, these cells, these viruses within the cell are about 150 nanometers. So a meter, a thousandth of a meter is a millimeter. A millionth of a meter is a micrometer. A nanometer is 1 billionth of a meter. So let's go back to our little scale here. There's me. There's our phytoplankton. To get down to the size of our virus, we're going 1,000 times smaller. So from me to this, 1,000 times, 1,000 times 13. This virus is 13 millionths of my size. Those little COVID viruses are about the same size, and man, something 13 millionths of your size can still make you ill. Isn't that stunning? Whew. So we've got these viruses which infect, and they can kill cells, and they do. And so there's another thing out there. The thing is, viruses are so small that we are not even able to see them with normal light. So how do we even begin to look at those viruses? Because they are so small, they are smaller than the wavelengths of visible light. So how can we see them? Now, the analogy I can give you is our vision is based on light. We can think about things being small and unable to see in the same way that we can think of things being far away and hard to see. So I got a question for you. How far can you see? What's the furthest thing away that you can see? Suggestions? Your vision's better than mine. You're younger. Any thoughts?
It says, I can be a clown and say the sun.
Go, Gavin.
How far away? Excellent. How far away is the sun?
That I have no idea. Let me check.
Anybody know?
Leora is supportive of Gavin. She says super far.
Super far.
There's basic head scratching in the room. Nobody seems to have a number.
Well, it could go for 93 million miles. That's pretty far, huh? Can anyone beat that? You're on the right track. I don't think that's a silly answer at all. I think it's a great answer. Can you see things for
other stars?
Other stars. I'll bet we can go in terms of light years for stars that are hundreds or thousands of light years away. Now, why do you see them?
Because they're bright.
Ah, they're bright. They're producing light. So even things that are practically way, way too small to see, you can see if they're producing light. Okay, makes sense? So if we could get a virus to produce light, we could see it, even if it were way, way too small to see. Does that make sense? That's the way scientists sometimes think. So here's what we're going to do. I get this dye. This is a fluorescent dye. It's called Cyber Green 1. It's complicated, lots of little ring structures here. But here's what happens. It sticks to the DNA that's in a virus. So I'm going to take a bunch of water. I'm going to filter it for these planktonic diatom viruses, catch them on the filter, and I'm going to add this stain. It's going to stick to their DNA. Now, here's the property of this dye. When I put blue light on that filter, it's going to be absorbed by this molecule, and this molecule is then going to re-emit green light. I put blue light in, it glows green. I can make it glow. And the green light that comes out, I'm going to be able to see, even though that thing is way, way too small to see normally. And that's how I'm going to see viruses. So I'm going to take a tablespoon of water from Lake Superior or Lake Michigan. I'm going to filter it. I'm going to look at that filter stained with cyber green under blue light. And what do I see? And here's what I see. Wow. Looks like a starry night. That teaspoon of liquid has about 5 million viruses in it. Perfectly clean water, but it's absolutely jam-packed with viruses. And at least some of those viruses won't do you any harm, but they're going to infect diatoms. That's kind of scary if you're a diatom, huh? Wow. So they're out there, and they're dangerous to diatoms. Getting old. Now here's a really interesting question. Here's a diatom. Diatoms reproduce by dividing by something we call binary fission. That means this diatom is going to divide in half. Now, when it does that, we've got two daughter cells. And at the point where that diatom divides, here's my question for you. How old are those new cells? So if this cell keeps dividing, does it keep resetting its age to 0? And if it keeps dividing, does that mean that this organism can reset its age indefinitely, which means if it keeps dividing, is it immortal? How's that for an interesting biological question? In my lab, I have a diatom which was pulled out from one single cell isolated from the bottom of a fjord in Norway in 1928. That one cell has been dividing continuously 2 to 3 times every day ever since. That's my candidate for an immortal organism. And I've still got it in my lab, and it's now all over the world in a lot of different labs. How about that?
And Gavin says, let's make our cells learn to become immortalized. And Leora says, how old was the original?
Well, that is a great question, isn't it? How do we age a diatom cell? How do we know how old it is? And is it regenerating itself every time it divides? I don't know the answer to these questions, but here's an interesting thing. If I were to take one of your cells from your body and try and grow it, it wouldn't do this. It would age and die. There is only one type of cell that keeps dividing and keeps dividing and keeps dividing. And it's not a good thing. Does anybody know what type, what general class of human cell does that? Most people don't want those cells in their bodies. Any thoughts?
Yes, it's come into the chat, cancer.
Cancer cells do that. Now, if we understood why diatom cells keep doing this, our cells don't, but why cancer cells do you think we might gain some insights into why cancer cells are so strange? Well, I do. And someday I'm actually going to convince the medical research community to study diatoms more. Huh? I've actually got some medical research money to do this sort of thing, but not a lot. But yeah, I will get there someday, because I really think there's a lot of interesting biological insight that might derive from this. Okay, last thing I want to talk about then, running out of something. So here is a happy, healthy culture growing along nice and green. And if I run it out of a nutrient, this happens, it dies. So would be immortal, but now I've run it out of something and here it's died. And typically the nutrient things it runs out of could be light, could be nutrients like the silica. So how do I study this? Well, it would be nice if I could measure silica in the cells. How could I measure silica in the cells? I'm going to show you one of my students measuring silica in the cells. So here's, oops. So I'm going to take you into my building, Lapham Hall, into my lab, and this is one of my students, Loretha. She's wearing a nice Diatom t-shirt, by the way. And Loretha's got a culture of cells, diatoms, out of Lake Michigan. And down she goes to the lab. And in the lab, a little bit of a mess, but that's the lab. She's got a microscope. And what she's going to do is she's going to take out some of those cells, and she's going to add a stain, which is specific to silica. It's going to stain all the silica. And that silica-specific stain is going to allow them, under UV light, to glow a nice pale green. And so everywhere there's silica in those cells, we'll see green. And the dye has a horrible name, which we abbreviate PDMPO. We put a camera on it, and all of a sudden we see glowing diatoms, and everywhere you see that pale green, you will see there is silica. Okay, and what you notice is some cells glow nice green and other cells in the chain don't. So some have silica, some are deficient in silica, and these may be on their way to dying. And aren't these pretty pictures? I really love these. Incidentally, under this light, the chlorophyll in these cells glows a nice red. That's one of the photosynthetic pigment. And so we get a whole array of this, but this gives us a tool to look at silica in these organisms. Now, we can look at the silica deficiency, but how do we know whether a cell is dying or not? Well, here's a nice healthy cell. It's got some chlorophyll, the photosynthetic pigment in it. And here's another cell. And when that cell begins to die, certain changes can happen in its cell membrane. And early on, those changes have to do with changes in the fats, in the lipids in the cell membrane, and we can label those with another dye. And that dye has this name, annexin 5, and that will make this cell glow. It glows red because of the chlorophyll, but it'll glow green because of the annexin. And we can measure how many cells are glowing green or red and green using something called flow cytometry. So flow cytometry allows us to count individual cells. In this plot, each little dot is an individual cell we've counted. On this axis, we have the amount of red chlorophyll pigment in them. On this axis, the amount of green fluorescence. So we have a little cluster, a clump of cells here, and they all have red fluorescence, but they have really low green fluorescence. And if you looked at them under the microscope, they're all glowing red, but there's no green. So these are all healthy living cells. Contrast that here. Here's a group of healthy cells, but here now we have cells that are just as red, but now have this dye attached. These are dead cells. And so we can differentiate between living and dead cells. Under the microscope, they look like this. They've got the red, but they've also got this green dye. So there's the way we can tell living and dead. So I mentioned a flow cytometer. What does that look like? Well, here's another of my students with a flow cytometer. Fancy looking instrument, huh? It's got a laser. Isn't that cool? Well, we're scientists. We use lasers. And it's going to draw samples in here into a flow cell, fire a laser at them, and based on the blue lasers, fluorescent emissions in different spectra, green or red fluorescence, it'll sort out those individual cells. It can count 10,000 cells in a minute. And so Chang Jae, who is one of the best students I've ever had running this machine, will put a small sample. The machine will suck it up. It'll draw the cells in, fire the laser at them, and count them. And in real time, you can see we've got some living cells here, we've got some dead cells here, but we can count them up and get thousands of counts right out of Lake Michigan really, really quickly. So that's how we do it. And this instrument, it's a little clunky. I've got a smaller one I can take on board a ship, and we can do the same sort of thing.
So John,
Okay.
just wanted to let you know that you've got a couple minutes, so hopefully we can get some quick questions in
All
for
right,
you.
and I'm just going to finish up right there, just by pointing out that the work we do that I've described is largely funded by grants we get from the National Science Foundation and also from Wisconsin Sea Grant. Okay, super. And I'd be very happy to answer any questions.
Not getting a ton of things. My first thought is the silica digested and used as a nutrient in anywhere? Is it just recycled then for the teeth and then back into the system?
Okay, so we don't actually have a whole lot of silica in us. We do put it in our bones, and so organisms like fishes will take a tiny amount of that silica out, but it turns out that in the lake, most of it does just get digested, recycled, and so we basically the organisms would excrete it. They'd pee or poo it out into the lake and it would get cycled around again. But we do use a little bit. We need it. It's a necessary nutrient, but not a lot.
We had a basic food chain question about if a fish is bringing it in, and then of course we're eating fish, are we getting silica?
You sure are. Yep. Probably even more important, though, something diatoms are very, very good at producing is something called omega-3 fatty acids. And fish are really good for omega-3 fatty acids. It helps your nerve development. They call fish brain food because of those fatty acids. The reason fish get them, though, is because diatoms produce them. And so we eat fish. We'd probably be as far ahead eating diatoms, although I will tell you, diatoms are super small, and I don't think they're particularly tasty. So I'd go with fish myself.
And then a question in the chat, what happens to the dead cells? Do they waste away or do healthy cells absorb them?
Right. So what we've seen, the vast majority of the cells that die seem to decay in the water. And so they break apart, they release all their nutrients back into the water. And so they really almost completely disappear. Some of them, and the silica shells in particular, fall to the bottom. And so they end up in the sediments. And in some areas of the world, those sediments become really, really rich in diatom shells. And we call that diatomaceous earth. You may see that in stores. We sell it, for example, you add it to your garden, and those silica shells will actually prevent insects from infesting your garden.
And thanks so much, John, for your time today and sharing with the students. I want to let you know that many have said that's really cool, your comments during the chat today. The class has ended, so we probably won't be generating more questions for you. But for those that are joining us from home, if you do have additional questions, we can stay on for you for a few minutes unless you're headed to another class as well. So if you do have additional questions, feel free to reach out to Mr. O'Neill, who can pass them on to either me or Dr. Berges directly, and we'll be happy to try to answer them for you. And we appreciate your time today.
Yeah, I'd
Cool.
like to really just echo that and say thank you very much. I found it really intriguing. The scale part totally blew my mind. And again, I always thought of, diatoms as just a little bit of floating glass. I don't really think of them as organisms per se. So thank you for changing my perspectives. Really appreciate it.
Excellent.
All right, I got some freshmen rolling in. I'm going to leave the meeting. So thank you very much.
Thank you.
Nice to
Mr.
meet you
O'Neill.
guys.
Nice to meet you. And thanks again to you, John. Just a quick follow-up. We will be sending the recording to Mr. O'Neill, so those that weren't able to join us, his classmates can, the classmates can see it. Thank you for joining us for today's Scientist to Students presentations, Great Lakes Research and Sharing, hosted by the University of Wisconsin Sea Grant.